Welcome to week 6. So this week we are focusing on the Streptococcus and Enterococcus group of gram-positive cocci. So they are gram-positive cocci and they are very much catalase negative. So last week when we discussed Staphylococcus, we learned that catalase is the first test we run after we see we have a gram-positive cocci. Of course, that's an aerobic or facultative anaerobe gram-positive cocci, not just a true anaerobe. But those normal gram-positive cocci that we see growing on our culture plates, we always run catalase afterwards. And that's because catalase helps us differentiate between the Staphylococcus group from the Streptococcus group. And so Streptococcus are very much catalase negative. Enterococcus genus in general is weakly positive. So it's kind of like the in-between. And then as we learned last week, Staphylococcus is very much catalase positive. So again, catalase is a test you perform in the lab with hydrogen peroxide as a reagent, and it's looking to see if these bacteria have that catalase enzyme, the ability to break down the hydrogen peroxide into oxygen and water. When it breaks it down, it bubbles with the oxygen appearing. So Staph are definitely bubbles, catalase positive, whereas Strep are catalase negative, and Enterococcus is kind of a little bit in between. So again, they are gram-positive cocci. They typically occur in chains. There is one strep that we will learn that likes to be in pairs. Staphylococcus, as we learned last week, was gram-positive cocci in clusters. So there's a little bit of a difference there on the gram stain. So this is what strep might look like on the gram stain. You can see the chaining effect it can have. This is a really long chain. They don't always chain that long. But you can kind of see what it means by saying it's in chains. And then here is the one strep that loves to occur in pairs, what we would call diplococci, diplo for two. So it is hard to see when it's just a still image like this. On your microscope, you would, of course, be able to zoom and focus in and out. But there's little gram-positive cocci in pairs, each one. So each one of these is an actual pair. It's not just one big rod or something. So they love to occur in pairs, and that's very much a unique feature for that strep. So streps are actually categorized on something that we call Lancefield groups. This is based on the carbohydrate antigens that they have on their cell wall of that bacteria. So there are definitely groups that will have a certain Lancefield group name assigned to them based on those carbohydrates. And then there are streps that don't, that are just not known as a Lancefield group, that they do not have those carbohydrates. But it is important to be able to interchange a Lancefield group name with the organism name. It's very common to refer to it either way. So the groupable streps, we have strep A, B, and D that are frequently seen in the lab. C and G are less frequently seen. And then the non-groupables, the ones that do not have the carbohydrate antigens, would be strep pneumoniae, and an entire group of other streps that we call just viridin strep. This is a bunch of different streps that might fit into that viridins group. They're a little bit, you will definitely see them, but just it's just such a big group of different ones that they just kind of group them under that name. So there's one example here like strep mutans. Okay. The other thing to note about streps is once we do the gram stain, it's a gram-positive cocci, and we catalase tested it and it's catalase negative, we're already thinking maybe we have a strep, or maybe it's an enterococcus and it was just negative or weakly positive catalase. So we're thinking strep or enterococcus. It's really important to look at the hemolysis reaction next, especially because it guides what test we're going to run next, because different hemolysis will be different tests that we would want to perform. So you definitely want to see if it's alpha hemolytic, beta hemolytic, or gamma hemolytic. And we did already learn these hemolysis patterns. But again, alpha hemolytic is a partial lysis, so it's going to have more of a greening color. That would be the center little diagram there. Beta hemolysis will be a complete full lysis around that colony. So that would be the bottom one is showing clearing. And then gamma is just no hemolysis at all, so non-hemolytic. So again, you would hold up your agar plate kind of up to the light to look through to see what that lysis would be. So based on the hemolysis will help guide what your next step in the flow chart system of testing is. So if we look here, this would be a beta hemolytic strep. You can see the full clearing around the colonies there. It ate away all of that blood agar, it lysed those blood cells in that agar around those colonies to make it clear appearing. And then here, this is a little harder to see, but it definitely is alpha hemolytic. If you look kind of over to the edge, you'll see that greening color. It's not as easy to see right here, but these ones right here, these isolated colonies here show a nice greening color around it. So just a partial lysis of those blood cells in the agar. All right, so the hemolysis goes with groups A, B, and C. They tend to be beta hemolytic. Group D tends to be either alpha or gamma hemolytic. Strep pneumoniae and the viridin strep are very much alpha hemolytic. And then Enterococcus likes to be anything. It can be any hemolysis. It just depends on the species and the strain. And of course, in a perfect world, these all would sit right where they're supposed to be. There's always once in a while a strain that'll pop up of something they're like, well, that should have been alpha hemolytic and it just didn't show the alpha like normal. But again, for the most part, they tend to stick to these hemolysis patterns. So let's start with our first strep organism called Streptococcus pyogenes. This is a very common strep seen in the microlab, a very big pathogen out of the strep. So this is what we would call group A strep. Sometimes you see the initials GAS to stand for that group A strep. But either name, whether you call it strep pyogenes or group A strep, same thing, very much beta hemolytic. Now, streps in general are going to be smaller colonies than the staphs. Last week on the staphs, when we started learning about them, we saw they were more medium to large, kind of creamier colonies. Streps tend to be smaller colonies, small to medium. And then again, you look at the hemolysis. So strep here, pyogenes, beta hemolytic. It is not typically normal flora of our body. We can carry it. It can colonize, but we're not usually having it as normal flora. So it a lot of times is carried in the upper respiratory tract, but it's always significant if it is found there because this is the guy that causes strep throat. So we've all heard of strep throat. This is the main culprit of strep throat is strep pyogenes. You can have sore throats for other reasons, of course. But when you hear of somebody testing positive for strep, this is what they tested positive for was strep pyogenes. So again, that's why it's clinically significant if you do find it carrying up in the upper respiratory tract. It's going to cause an infection. And then it can colonize our skin and throat, and that will help make its source of transmission for it. But again, not normal flora in any way. So the virulence factors, it has quite a few virulence factors to make it really pathogenic, to make it really good at causing infection. The main one in order to even produce virulence factors is the M protein. It has to have the M protein in order to have the production of virulence factor. It also allows it to be resistant against phagocytosis. So again, your body, your neutrophils in your body are going to try to gobble up this bacteria, get rid of it, but the M protein kind of helps allow it to resist that phagocytosis, those neutrophils, and then it also helps it produce these other virulence factors. Streptolysin O and streptolysin S are what will help make it beta hemolytic on the blood agar, so they will lyse blood cells. So I always think of the O for drilling holes in the agar, S kind of clears out the cells then once it drills the holes into the agar. That's the way I think of it. Hyaluronidase is an enzyme. It's called the spreading factor. It is an enzyme that kind of helps the bacteria get through into your connective tissues and spread in your body. So when I think of spreading factor, I just think of it's really spreading in your body, digging through those tissues in your body. The way that I remember hyaluronidase as a spreading factor is I think of hollandaise sauce. I don't know why, but that sticks in my brain. And I think if you get what is it, eggs Benedict, which I don't even eat, but you have hollandaise sauce on that and it spreads out through the plate. If it's a sauce, it spreads on the plate. So I think hyaluronidase spreads. DNase is an enzyme that just allows it to break down DNA of cells that are going to try to fight against the bacteria. And then it produces a lot of other exotoxins. There is one exotoxin specifically that will produce the rash that's seen as scarlet fever. So strep pyogenes is responsible for causing scarlet fever. It's not something we hear about in modern times, but it's definitely something that we've seen in movies that reference, years ago, 100 years ago or so. So there is a rash that's very commonly seen with scarlet fever. And the toxin that strep pyogenes produces is called the erythrogenic toxin. And I can type that out here, erythrogenic toxin. And that's the toxin that's responsible for the rash. So when you see the word erythrogenic, erythro means red. So red, scarlet. So that's kind of how I keep that tied together in my brain. And then there's also other toxins it can produce. This also can do toxic shock syndrome. We discussed that with Staph aureus can do that as well. So another one is strep pyogenes. It has another exotoxin that can produce toxic shock syndrome too. So lots of potent stuff that it can produce to cause infection. All right, so diseases, a lot. This is just some. You know, there's way more that can go along with strep pyogenes than this, but probably the most common one, well, not probably, is the most common one is strep throat, or you can just call it pharyngitis. So we definitely don't really culture strep pyogenes as, I mean, we do culture for it off of strep throat, but usually when somebody gets their throat swab, that swab is now just being tested by PCR methodology. We're looking for DNA to say, yep, strep pyogenes there, nope. You can also do a basic kit. There's still definitely those basic strep kits that can be done on the bench top that just take a couple minutes. And then if it's negative, a lot of times they'll still set up to a culture plate to confirm that it was truly negative, because sometimes strep pyogenes might pop up, cause a negative on the kit, but might come up positive on the culture instead. Most labs have converted to running PCR molecular methods to look for strep or not. It's a much more accurate methodology. It's still fast. It takes 10 minutes, you know, and they still get an answer. And then because it's more accurate, you don't have to like culture it always, but it depends on what your microlab decides. A lot of times they might still set up culture plates for all those negatives just to verify that it was truly negative. So that's strep throat, pharyngitis. Like I mentioned, scarlet fever. Necrotizing fasciitis is that whole flesh-eating disease. You've heard of probably news articles, extreme cases where they get the flesh-eating bacteria, strep pyogenes. There are strains of strep pyogenes that can do that. Most strains do not, but there definitely are strains that can. Impetigo is like a skin infection. It's a lot of times occurs next to the mouth area, especially in young children. Cellulitis is a very, you know, more serious it's not on the surface, it's not skin level, it's like the epidermal level. So it's not like usually found on top. You might not even see you have a wound on the top surface, but it's occurring underneath, and it can become very septic easily, meaning it can get into the bloodstream and then spread throughout the body easily. So that's something cellulitis is treated very quickly. A lot of times with an IV antibiotic to really like try to get rid of it quickly, because again, there's risk that it could spread into the bloodstream. Staph aureus was also one that can cause cellulitis. So it's not just strep pyogenes. There's a bunch that can cause cellulitis, but this is one. Toxic shock syndrome, as we mentioned. And then there's these last two that are called post-streptococcal diseases, meaning you had an infection with strep pyogenes, say you had strep throat, and you know, you kind of, and it spread, or, you know, and it went to somewhere else. Or the toxins that it released go and cause damage somewhere else in your body. So that's what's meant by post-streptococcal, is that you have this infection, the strep pyogenes infection, in one part of your body. And then it spreads. I apologize, my dog has decided to bark. Okay, I'm hoping that I stopped him for a second. So anyway, so the toxins, you know, your body's response, whoever is, has also now damaged another part of your body. It could be the antibodies that your body released to fight that strep pyogenes with the antigens have now caused damage to something else. So two that are linked with this is rheumatic fever, which is issues with your heart. Rheumatic fever is different than scarlet fever. Just keep that in mind. But rheumatic fever is damage to your heart. Acute glomerulonephritis is damage to your kidneys. So the antibody antigens that, you know, you've kind of, the antibodies you made with the antigens combining of the strep pyogenes might damage those kidneys, you know, and cause damage to the glomerulus down in the kidney, something like that. So it does happen with strep pyogeny infections. They're called post-streptococcal diseases. All right, our next strep is strep agalactiae. This is known as group B strep, also very much beta hemolytic. So we just got done with strep pyogenes, which is group A and beta hemolytic. This is group B and beta hemolytic. So as you can imagine, when we get in the lab and we see we have a gram-pos cocci catalase negative, they're beta hemolytic, we would need to differentiate, do we have strep pyogenes or do we have strep A galactiae? So we will discuss testing that we can do to separate between which beta hemolytic strep you might have. So strep A galactiae is definitely one that's seen with neonates, so babies, brand new babies, infants, acquired it in the birthing process. So women can carry strep A galactiae, group B strep, in their birth canal. It can just be part of that, colonizing it. So one thing that's very commonly done in pregnancy is at a certain point in pregnancy, the provider will swab and have it tested to see if they are somebody that does have strep A galactiae. If they do, they are treated with antibiotics to help wipe it out before they give birth. And that way, upon giving birth, it is not passed to the infant. Otherwise, there is risk that it is passed to the infant and it causes neonatal meningitis, which is extremely serious and can cause the baby's death. So it is important to always have that screening done as part of pregnancy to look for that strep A galactiae. And strep A galactiae's virulence factor, one of its main ones, is it does have a capsule that prevents it from phagocytosis. So what I think here is, I always think group B for baby. So I link it to causing that neonatal meningitis. Group B for baby is how I always kind of think of it. Some other beta hemolytic strep, group C and G, now just considered one kind of single subspecies, Streptis galactiae, subspecies Equisimilis. Very similar infections to strep pyogenes with some skin ones, things like that. No associated virulence factors that have been heavily defined as of yet. All right, switching over to strep pneumoniae, another really pathogenic, prevalent bad boy. No Lancefield group with this name, with this strep. So it did not have any carbohydrate antigens on its cell wall. It is alpha hemolytic, and this is the one that likes to occur in pairs. So what we would call gram-positive diplococci. So the DC for diplococci, meaning it occurs in pairs. A lot of times these pairs have been referenced as cat eyes or lancet shaped. So cat eyes meaning that really super almondy kind of pointed end. I know it can be sometimes hard to see on, you know, an image that we saw earlier. It was hard to see there. You would really need to have a good microscope with focus to kind of see the little bit more of a pointier ends when it occurs in pairs. But just know, if you hear of a diplococci looking lancet shaped or cat eyes, they're kind of referencing the strep pneumoniae appearance pairs. The virulence factor here, this also has a polysaccharide capsule that lets it avoid being phagocytized. That is its key virulence factor. It's really effective. So definitely a pathogen. It is huge cause, as its name implies, of pneumonia. It is the number one cause of community acquired pneumonia. And then it also is a big cause of meningitis. So two very significant infections that it is known to be linked with. And of course, it's not just those two. We can find strep pneumonia with some other things, but we're just whittling it down to the essentials here. And then the other alpha hemolytic strep group is that whole viridin streptococci. So this also has no Lancefield group name to it, no carbohydrate antigens on the surface. And this is just an entire group of other alpha hemolytic streps. There's a bunch of other alpha hemolytic streps that can fit into this group. And so strep mutans, strep mitis are two more common ones out of the viridins group. They are opportunistic pathogens. They tend to be more low virulence, so they don't have a lot of virulence factors, so they don't cause as much infection. They very much just take opportunity when they can. When they have been linked to diseases, endocarditis was one. Dental caries, meaning like infections with dental cavities, is another prevalent one with viridin strep that they've been linked with. So that's the mention there. I think on the one side when I mentioned strep mutans, it said dental caries next to it on that one. And then non-hemolytic strep, the biggest one here would be group D streptococci. This is the Streptococcus bovis group, which includes Streptococcus galolyticus. So in the group D, it'd be Streptococcus bovis group. They call it a group. Strep galolyticus is one member of that group D. So most of the time they are non-hemolytic. Sometimes they might pop up as alpha hemolytic. They are commonly found in the gastrointestinal tract, especially of cattle and sheep, thus the name bovis, bovis for bovine or cows. Diseases that they're linked with, definitely UTI, so urinary tract infections, and also endocarditis. And then finally, enterococci. Again, enterococci can be any hemolysis, so alpha, beta, or gamma, just depends on the strain and what you have. Sometimes people call Enterococcus group D. I know we just got done discussing group D is strep bovis, strep galolyticus. Once in a while, you might still hear enterococci referred to as group D, but it truly isn't that group. It is widely associated with nosocomial infections. So nosocomial means, again, hospital-acquired infections. UTIs especially. So hospital-acquired UTIs is the main infection source for enterococci. The two most common species out of here is going to be Faecalis and Faecium. Enterococcus is coming out of our GI tract. So that's kind of where it's found as like normal flora. If you look at the name entero medical terminology wise means intestines. So GI, gastrointestinal. So it is a normal flora of our intestines. It is also the very first gram-positive cocci that was found to build resistance to the antibiotic vancomycin. So there are definitely species called vancomycin resistant enterococci. So that is important, and we want to make sure that doesn't become more prevalent. Of course, we discussed antibiotics. Last week we discussed MRSA, methicillin-resistant Staph aureus. We also have this VRE. You're going to learn that more and more of these bacteria become resistant to drugs. As they're treated, they learn how to adapt and get around them. So this is the first gram-positive cocci that built resistance to vancomycin. All right, so now that we kind of covered some of the more main pathogens out of the streps and enterococcus, let's discuss diagnosis. So of course we have, as we mentioned, like kits, strep kits for antigen detection. So strep pyogenes antigen can be detected on throat specimens using an ELISA method kit or latex agglutination kit. These are methods that you can learn in immunology class that's in the kits. So they're looking for that strep pyogenes antigen, and they have antibodies there that will bind with it, produce a color, change in the kit that say it's positive, something like to that effect. Again, if it is negative on the kit, a lot of times we still set it up to a blood agar plate, culture it, incubate it, and look to see if it's prevalent, because there are times that it's negative on the kit, but it still might be positive on the culture. So oftentimes what it was done is when somebody's throat would get swabbed, they get swabbed with two throat swabs. One goes for the kit, one other swab goes for the culture plate. Now a lot of people have switched again over to the PCR methodology. There are kits available also for strep A galactiae. So again, you can use a kit to look for strep A galactiae, like during the pregnancy, if you're swabbing to see if they carry it, you might be using a kit there. Of course, gram stain, we always start with gram stain when we're looking at culturing anything. This again will be gram-positive cocci in chains. As I said earlier, strep pneumoniae is the one that's in diplos and pairs. So we call those lancet-shaped or cat eyes. Sometimes you might see them in short chains, a lot of times in pairs. And then again, as soon as we know it's a gram-positive cocci, we would catalase test it. So this again, streps are going to be catalase negative, enterococcus catalase weak positive. All right, media of choice, sheep blood agar will support the growth of all of these, chocolate agar as well. MacConkey, no, MacConkey only grows gram negatives, so that will not work for streps and enterococcus. CNA and PEA, very much yes. Both of those, remember, are plates that will grow gram-positive organisms, so both of those would be great choices as well. So a lot of times we definitely always set up a sheep blood, chocolate agar. It depends on where in the body, the source of the infection that we're setting up to. But CNA and PA both will grow. For group B, if we're looking to see if somebody has that strep A galactiae group B strep in their birth canal, swab, we can use a special broth called a limb broth, and that is specifically used for this. So you can, kind of swirl the swab in that limb broth, incubate, and look at it the next day. Limb broth has different has different antibiotics. Oh, I couldn't think of the name, and it's right under there. It's right here. That will help suppress the growth of all the other normal vaginal flora that might be in there. And you have a lot of normal flora in your vagina, so we'll have antibiotics added in that limb broth to kind of suppress the growth of the other normal stuff. That way, if there is group B strep there, it will come through, and we can, look for it that way. So limb broth is used for the group B strep. Incubation. Streps and Enterococcus love carbon dioxide. They need carbon dioxide in order to grow thoroughly. So we would want to make sure that we put these in a candle jar, is commonly used, maybe some sort of CO2 incubator, something of that nature. We want to ensure that we have enhanced or increased carbon dioxide levels to help them thrive. If we are specifically looking for the beta hemolytic streps, like strep pyogenes, strep A galactiae, they a lot of times you might see stabbing on the agar plates. So it's showing you here the stab marks here, or kind of in this diagram. That is just because it's enhanced by anaerobic conditions. We call it a facultative anaerobe. Those stabs really allow that beta hemolysis to be enhanced. So if the beta hemolysis is there, they really show up in the stab marks. So you can see that in the bottom picture of how enhanced versus the colonies. Around each colony, the beta hemolysis might be not as well seen, might be a little bit dimmer. Whereas with the stab marks, it really enhances the look and appearance of that beta hemolysis. So if it has beta hemolysis, they will show through in those stab marks. Okay, so now I said, once you catalase, you Gram-Stain catalase tested, and it's catalase negative, we pause. You always look at the hemolysis next, and your hemolysis determines what groups of testing you should run next. Beta hemolysis, we love to run a bacitracin, what we call A-disc. CAMP test could be performed, hippurate, PYR. Alpha hemolysis, it could be an Optekin P-disc, biosolubility. Gamma, or anytime you suspect Enterococcus, could be 6.5% NaCl, bioesculin, biosolubility, PYR. So there's a couple of repeat tests in a couple of these. But again, this really guides our next set of testing in the flowchart system to help differentiate between what could be occurring. So the bacitracin A-disc is literally a disc you put on the blood agar plate. You streak out a lawn of your organism that you are suspecting. You put the A-disc on there, you incubate it, and you look at it the next day for a zone of susceptibility. If there is a zone around that A-disc, and again, you usually measure the zone to see if it's a certain millimeters, you can call it susceptible. If there's a nice zone around that, that meets the millimeters. If the bacteria grows right up next to the disc, that would be called resistant. So again, bacitracin is done with the beta hemolytic strep. So those are your strep pyogenes and strep A galactiae. Strep pyogenes is very much bacitracin susceptible. The reason it's called the A-disc is it's looking for strep pyogenes, which is group A strep. So that's why they call it the A-disc. It has the letter A on it. It is looking for group A strep. So strep pyogenes is very much bacitracin susceptible. Strep A galactiae is bacitracin resistant. So you can see how that test alone helps separate which beta hemolytic strep you might have. There is a CAMP test you can run. CAMP test is also just performed on a blood agar plate. You actually take a streak of Staph aureus. Right here would be Staph aureus streaked across the blood agar, which Staph aureus, again, remember, is beta hemolytic as well. So you streak that Staph aureus across, and then you streak your suspected organism down to meet it. So if we're looking for, strep A galactiae or strep pyogenes, a beta hemolytic strep, so you streak that here, whatever is our suspected organism, and then you incubate this, and the next day you look to see if there's like a triangular formation between the two streaks. So you can see they didn't streak it to touch. It's just right before that. So again, you're looking for that triangle formation of beta hemolysis. If that triangle beta hemolytic is there, it is positive. So strep A galactiae is very much a positive CAMP test. Strep pyogenes is negative. So again, this is another test that helps separate between the two beta hemolytic streps. So strep A galactiae, positive CAMP, strep pyogenes, negative CAMP. All right, switching over to Optekin disc testing. This is to help with the alpha hemolytic streps. Again, it is a disc. So same thing, you streak out a lawn of organism on a plate, you put the P disc down, you incubate it, look at it the next day for a zone of susceptibility. If there is a zone, it is susceptible. No zone, if it grows right up to it, it is resistant. So Optekin is looking for, the zone of susceptibility is looking for Strep pneumoniae. So there is a P, it's called the P disc. There's a P on it for pneumonia. Pneumonia starts with a P, even though it's silent. So that is why it's called the P disc. It is looking for Strep pneumoniae. So Strep pneumoniae is very much susceptible to Optekin, whereas all the other alpha hemolytic streps, the viridins group of streps, are resistant to Optekin. So again, this is an alpha hemolytic test for strep. So Strep pneumoniae is susceptible. The rest of the alpha hemolytic streps, the viridins group, is resistant. You can also do bile esculin testing. This can be done more for when you suspect Enterococcus, but Enterococcus, again, can be really any hemolysis. So this is looking, there's a couple ways to do bile esculin. There's also a tube form of bile esculin in a glass or plastic tube, but this is showing it on an agar plate. So you can have it either way, but it is looking to see if it thrives in the presence of bile. Enterococcus are very much bile esculin positive. They thrive in the presence of bile. All right, so it is a lot of different tests to figure out. And so there's definitely a flow chart system here. So again, I said gram-positive cocci, catalase negative. If you think it's beta hemolytic, then you could do your bacitracin test. Susceptible is definitely that group A, strep pyogenes. If it's resistant, you can go further and CAMP test it. Positive would be that strep A galactate, group B. If it's negative, maybe it's one of those other beta hemolytic streps that we didn't really go too far into. Maybe it's group C or G. Maybe it's that group D that once in a while can show up as another hemolysis. So you can see how that just kind of helps start to separate between the beta hemolytic streps. I should go back up here. There was a couple others I mentioned. Hippurate, strep A galactiae is also positive for hippurate hydrolysis. Strep pyogenes, negative. PYR testing, strep pyogenes is positive for PYR. Strep A galactiae is negative. So if we actually detail this out, which I don't know, I don't know why it's being funky there. Okay. So strep pyogenes susceptible, strep A galactiae resistant. And then CAMP, again, was strep pyogenes. We'll just keep it in the same order here. Negative, strep A galactiae positive, hippurate, strep pyogenes negative, strep A galactiae positive, and PYR strep pyogenes positive, strep A galactiae negative. So at this point, you kind of just have to start memorizing what reactions go with which, organisms. And that's just, unfortunately, the problem with microbiology is there's so many organisms, there's so many tests, and it's a lot of memorization. So that is those test results there for the beta hemolytic streps. If you want to differentiate alpha hemolytic streps, again, it's gram-positive cocci, catalase negative. Once you see you have alpha hemolysis, you could run the Optekin disc. Again, susceptible is very much strep pneumoniae. If it's resistant, could be a viridin strep, could be Enterococci. If the Enterococci is coming up alpha hemolytic, so you could dig in. You could maybe do a bile esculin. Bile esculin positive is definitely Enterococcus, but it also could be group D strep. Bile esculin negative is viridin strep. If it's bile esculin positive, maybe run a 6.5% NaCl or a PYR. Enterococcus is positive for both of those. Group D strep is negative for both of those. And again, your group D strep is your strep bovis galolyticus whole grouping that can be gamma hemolytic or alpha hemolytic. So again, there's lots. It would be easy if these organisms always kept the same hemolysis, always behave the same way, but they don't. So they can fall under a couple categories at times. So this is a good testing system to kind of start separating out between them. So again, if we go back up to this slide here, so under the alpha hemolytics, we already have discussed very much that Opticum P. disc, strep pneumoniae, I'm just going to put pneumo, is susceptible. The viridins group and enterococci, if you want to add that in here, are resistant. Biosolubility is different than bile-esculin. Biosolubility is, you can do it actually right on the agar plate. You can add a dropper of that biosolubility reagent, and you can determine if it solubilizes the colonies on the agar plate. Strep pneumo is positive for biosolubility. It does, its colonies will become soluble under that reagent and kind of clear away. Whereas viridins and enterococci are negative for biosolubility. Again, biosolubility very different than bile-esculin. All right. And then the gamma, anytime you suspect enterococcus, or you're trying to figure out if it's enterococcus or group D strep. I love that my dogs are just, they're going crazy, of course. I apologize if you can hear that in the background. Okay, let me just raise this up so I can type here. Okay, so again, 6.5% NaCl is, so it's looking to see if it's tolerable in salt. Can it grow in salt? Does it like to grow in the presence of salt? Enterococcus loves to grow in the presence of salt, so it'll be enterococcus positive. Whereas group T strep is negative. It does not like to grow in the presence of salt. Bile-esculin, again, that's if it loves to grow in the presence of bile-esculin. Enterococci is positive here. Group D strep is also positive here. Both will like to grow in the presence of esculin. If you think it's one of the alpha hemolytics, those are negative. So strep, pneumo, and viridins are both negative for bile-esculin. And then finally, biosolubility, we already said that up above. PYR, enterococci is positive, and group D strep is negative. So that will help separate between those there as well. Okay, so lots of testing here. And again, this last flow chart is referring to the non-hemolytic. So again, you can do bile-esculin, you can go over to 6.5% to separate out. And so we just kind of reviewed that together. Okay, that is your week 6 streps. Let me, 1 second here. I feel like I had some other PowerPoint slides. 1 second, let me look for something. Okay, never mind. I thought I had something else on there, but nope, I have everything on there. I think the hardest part again of the streps is going to be an enterococcus is how to differentiate between all of them, because there are so many different groups of tests. So very much try to memorize what hemolysis each strep belongs to and the tests that we use for that hemolysis pattern. That is the easiest way to start to organize your testing process. And, you know, I understand the gamma hemolytic one gets a little bit more tricky because, and then enterococcus gets tricky because enterococcus really can belong in a couple of it can belong in any of the hemolytic patterns. So again, enterococci, it has a couple unique features that differentiate from the others, like that 6.5% salt growth. None of the other strep organisms do that. So that helps identify it. Bile-esculin helps to a point. Group D strep does like bile-esculin, so that, you know, you have to keep that in mind. But there are a couple key tests that go with that enterococci. So start to really dig into those flow charting and testing systems with streps and enterococcus. And of course, your student lab will also help with that. All right, I hope everybody has a great week.